everybody. Welcome to our first mini lesson on the digestive system. There's going to be quite a few mini lessons in this system because it's a big system. If you've worked through your homework or just kind of flip through the chapter, you'll see it just kind of keeps going on and on and on. So again, I'm just going to be breaking down the topics via physiological processes. Basically, the way the book introduces it, it kind of goes through the alimentary canal, talking about the anatomy and the jobs, the physiology of each one of those parts as you progress through. I'm not going to spend a lot of time on the anatomy, so if you do have any questions over the parts and pieces part of the digestive system, send emails, stop by a Zoom office hour, whatever you need to do. So I'm just going to focus on the physiology, kind of the functioning of the digestive system with the understanding that you know or are familiar enough with the anatomy. So if you're not there yet, make sure you go and look at the anatomy pieces before you start jumping into the physiology. So this little section, this lesson, I'm going to cover the layering, the histological organization of the alimentary canal, and then the control of the alimentary canal. So both of those topics are presented early on in the chapter, which are kind of the foundations, some of the foundations for when we get into the anatomy and the physiology in later ones. Before we get into those, I am going to talk about, usually what I do in class is I talk about the big picture. Because this is such a huge chapter, there's lots of parts and pieces to it, it's easy to get lost within the minutia, if you will, of your whole, 30-foot long digestive tract. So the big picture and the reasons why all of these parts and pieces are here is to absorb nutrients. That's kind of the main goal, and to get rid of the things that we can't absorb. So we take in food into our digestive system. Somehow we have to break that down into the small molecules that are able to absorb across our digestive epithelium to get into our bloodstream. And then anything that we don't use, we get excreted. So every single part of the digestive system is there to accomplish that goal. So when you're working through your different body parts of the digestive system parts, what does the mouth do? What does it look like? What is its structure? What is its function? What role does it play in getting the food molecules, these large chunks of food, down into those small molecules that can be absorbed across the digestive epithelium? What does the esophagus do? What does the stomach do? What does the small intestines do? How does the liver help? The pancreas, the large intestine, all of those parts have a role to play in this big picture. So I wanted to just remind us what those things are that we're working to absorb. So if you remember our large major macromolecules, carbohydrates, you might remember those, carbons, oxygens, hydrogens. These are things like starches and sugars and polysaccharides, and they're building blocks. So what carbohydrates are made out of are monosaccharides, right? So remember that. It is the monosaccharides that are small enough to be able to be absorbed across the digestive epithelium. So no matter what kind of carbohydrate you ingest, the ultimate goal is to get that down into a small monosaccharide like glucose or fructose or galactose, which then can be diffused, facilitated diffusion across that digestive epithelium. Okay. What are some of our other macromolecules? You guys remember proteins? Right? So proteins, big, large, very complex molecules, and what is their building block? What are we trying to break proteins down into? Amino acids. So again, proteins from dairy or meat or eggs, whatever you are consuming that has these large animal or plant proteins in them, our job of the digestive system is to break them down into individual amino acids, because it's at this stage, these individual building blocks, that we can absorb across the digestive epithelium. All right, what's another one of our macromolecules? Lipids, you guessed it. So lipids are fats. And so there's a whole bunch of different kinds of lipids. I'm going to focus on the nutritionally important lipids, which are your triglycerides. And triglycerides are made up of fatty acids. So again, the food that you're eating that contains lipids, your body's goal is to break that down. I should say your digestive system's goal is to break those lipids down into fatty acids and allow them to be reabsorbed across the digestive epithelium to be used by the body. And then our last major macromolecule is not so much nutrition based, but we still have to address it, are the nucleic acids. So remember, nucleic acids are DNA and RNA. and their building block was, drumroll, nucleotides. So A's, T's, C's, and G's. Now we don't use nucleotides for nutrients. What do you think we're going to use them for? Well, nucleotides build DNA and RNA. So if we are ever going to do mitosis and we're ever going to do DNA replication, which our cells do all of the time, some way more than others, we need that building block to be able to make our own new DNA and RNA. And where do we get those nucleotides? From the food that we eat. Okay. So this big picture, we have our major organic macromolecules, carbs, proteins, lipids, and nucleic acids. The goal of the digestive system is to take those big things that you eat in your mouth, and by the time we get down into our small intestine, we have broken them down into these smaller building blocks, these monomers, if you will, that then are small enough to be absorbed across the digestive epithelium. And now we have our raw material to make more of what we need for our body. And then anything else that's not absorbed is waste product. And then we have other parts of the digestive system to get rid of, store and get rid of that waste. Okay, so that's the big picture. So keep that in mind every time you're looking at a different component of the digestive tract. Think to yourself, what is the role that the oral cavity has to play in this big picture? What is the role the duodenum has to play in this big picture? What's the role the ascending colon has to play in this big picture? So if you kind of keep looping around to this big idea, hopefully that will get you a better understanding. Okay, so the parts that I want to talk about after the big picture here. All right, I had to take a pause real quick, so hopefully that didn't mess things up. So on this slide, I'm going to talk about the histological organization of the digestive tract, which is also known as the alimentary canal or the gastrointestinal or GI tract. So lots of different names referring to basically the continuous tube from mouth to anus. And I don't like to spend a lot of time on anatomy in these little mini lessons because I figure most people can get through pictures and stuff okay on their own. But this is really important to understand these layerings. And the reason why I wanted to spend some time on this is because the whole structure and function theme. So we have this basic pattern, these 4 layers that we're going to see, and they're pretty much the same 4 layers in every part of the alimentary canal with some modifications. So understanding the basics of these 4 layers and then understanding the modifications can help you understand the functions and why they are slightly different from one place to the next. All right, so we're going to first start with the outermost layer. And so this is what we call the serosa. It is also known as the visceral peritoneum. So as you're going through the beginning pages of this chapter, it talks about the mesenteries, the parietal and visceral. Hopefully you're familiar with those. Remember, parietal's on the wall of the cavity, visceral's on the surface of the organ. So when we're talking about this connective tissue sheath on the outside edge of any one of your tubes, the stomach, the small intestine, large intestine, we call this the serosa, also known as the visceral peritoneum. So it's just a connective tissue sheath. All right, the next layer is called the muscularis externa. So muscularis externa, the new edition might have a different name for it. It's just the outer layer of muscles. And there's typically 2 layers within this muscular externa. You're going to have the circular layer and the longitudinal layer. Okay. So circular is so if this is my tube going this way, circular layer, the fibers are going to be going in this orientation. Longitudinal layers, the fibers are going to be going lengthwise along the length of the tube. And when you read about peristalsis and segmentation, these 2 layers of muscle allow for the tube to squish kind of in diameter this way, and it allows it to shorten in this way. So that allows this peristaltic movement, this wave-like action to constrict and then shorten, moves the bolus of food down the tube in this one-way direction. So that's the job of the circular and longitudinal layers of the muscularis externa. The next layer in is called the submucosa, and it's primarily loose connective tissue. Think like a dermal, dense irregular connective tissue. It's going to be very vascular. It's going to have some nerve supplies in it. It's going to have some glands located in it. And then the innermost layer is called the mucosa. And that's your lining. There's some areolar connective tissue under there, and it's got the surface tissue. So since it's lining the opening of a tube, this lumen, it's going to have an epithelial component to it. And we're going to see that epithelium change as we go from one place to the next. So these 4 layers, serosa, muscularis externa, submucosa, and mucosa are these 4 layers of the digestive system histology that we're going to see in all of these different parts, from the oral cavity to the esophagus, stomach, small intestine, large intestine. And then we're going to see some modifications, and I'll show you some examples here in a second. So those are the 4 layers, but they're like, hey, Corey, there's a whole bunch of other stuff on the other side of this picture. What are all that? All of these things over here. So in reference to some details within each of these 4 layers, the mucosa right here is made up of the mucosal epithelium, which is going to be the epithelial covering. And then the lamina propria is like your areolar connective tissue. It's the basement membrane and some connective tissue. It also has what's called the muscularis mucosa. So this kind of goes with that. It is a thin muscular layer that the mucosa sits upon. Now, when we're talking about, say, the esophagus, that muscular or mucosal epithelium is going to be stratified squamous epithelium because you're still swallowing pretty large chunks of food. We need that thick tissue for protection. When we get into the stomach, it transitions from the esophagus of stratified squamous to a simple columnar, and it runs simple columnar all the way through the stomach, small intestine, large intestine. By the time we get to the rectum and the anus, it turns back to stratified squamous epithelium because we're getting back to solid waste material moving through. We want to prevent abrasions and damage. So think of the mucosal epithelium as primarily simple columnar epithelium through the majority of your tube. And then here's some glands. You might have some glands associated with the mucosa. Here's our submucosal glands. So these guys are going to be associated in that dense, irregular connective tissue layer. And then we're just labeling, there's some lymphatic vessels, there's some blood vessels, and then we have some nerve plexuses. So submucosal just means it's sandwiched between the submucosa and muscularis externa. The myenteric is nerves sandwiched between the circular and longitudinal layer. Okay. All right. So when we take a look at this histological organization, we're really trying to see those 4 layers whenever we identify a different, like a new part, whether we're looking at the esophagus or the stomach or an intestine. Can we find those 4 parts, and can we see the modifications to illustrate the function? So what I'm going to do next, I have some pictures of actual histological slides. I know we're not spending a lot of time on histology because we don't have the microscopes, and it's just kind of weird doing it in lab with pictures. I might show you some, but I'm not going to be as heavy-handed with the histology as we would have in a live class. But I still wanted to show you this for comparison's sake. So I have 4 parts of the alimentary canal here. And so we have up in the upper left, this is the stomach. This is the jejunum. This is the ileum. And this is the colon. Okay. All right, so when we're looking at the stomach, I tried to find pictures that actually had labels so you can see from that wonderfully drawn diagram on the last slide to actually what it looks like in real life. But we can see it. So in the stomach, so this outermost connective tissue here, that's going to be the serosa, just a thin little connective tissue sheath. And then here's our muscularis externa. And it's a little bit hard to see on that slide, but in our slides in class, we would be able to see the 2 layers, the circular and the longitudinal, and it looks like we'll be able to see that in the colon. And then here's your submucosa, this kind of flaky looking, dense, irregular connective tissue. This little guy is our muscularis mucosa, and then all of this is the mucosa. It's pretty thick, right, compared to the picture that we just saw. Now in the stomach, your mucosa is really thick, and one of the reasons why is because your stomach squirts out a ton of acid and enzymes, and we don't want that to be digesting the inner lining of your stomach. So you have a nice thick mucosa, and you have all these little pits right here. You see all these little pits. So they're called gastric pits, and they're lined by goblet cells that secrete a thick alkaline mucus, and that also helps to protect the lining of the stomach. And then all of this layer right here, all of this stuff, you can't really see it, those are the gastric glands. We'll be talking about those in the stomach mini lesson lecture. but they're the ones that are shooting out all the enzymes and the hydrochloric acid and some of this mucus. All right, so that's the stomach showing those 4 layers. So in the jejunum here, we can do the same thing. So here's our serosa, here's our muscularis externa. It's not quite as thick as the muscularis in the stomach. And it kind of makes sense because your stomach is a churning mixing bag, so we need a lot of muscle. By the time we get the food to the duodenum or the jejunum, the small intestine, we don't need as much muscle. So we're just doing peristalsis. We're not actually physically smushing the food. Okay. And then here is our submucosa with a whole bunch of glands. They're called Brunner's glands. I don't know if you're going to get too much into that. Here's our little muscularis mucosa right here. So that lets us know we're at the level separating the mucosa from the submucosa. And in the jejunum, we don't have a whole bunch of pits. We have these long kind of finger-like processes called villi. So each one of these guys right here, that's a villus. It's most like the diagram that we just got done going over. It's got simple columnar epithelium. This is the absorptive surface area. This is where the nutrients are coming across. And if we take a look at these villi, look at all of that surface area. So each one of those villi is covered by absorptive cells. These are your simple columnar epithelium. So it makes sense that our mucosa is going to be in all of these folds in the small intestine because that's where we're absorbing the nutrients. By this time, everything's been broken down to those monomers, the monosaccharides, the amino acids, the fatty acids, and now they're ready to be absorbed. So it makes sense that our intestinal lining is going to have all of these villi, the folds. And they're lined by the cells have microvilli. So even those simple columnar cells, they themselves have increased surface area. So the structure and function really match up really well with that. All right. So then down this on the bottom here, we have our ileum, bottom left. So our serosa is going to be out here. Our muscularis is very thin. So that's our muscularis mucosa. Here's our submucosa. And kind of right where I'm drawing that line would be our muscularis mucosa. And then this is our mucosa. So we see some villi. They're not as many and they're not as crammed. And that kind of makes sense because by the ileum is the very last part of the small intestine. So by the time we get to the ileum, there's not as much absorption needing to take place. It's all been done. So we don't need to put as many villi in the ileum as we do in the duodenum and the jejunum. But you might also be noticing something else. I'm going to circle these guys right here. What do you think those might be? Think back to the chapter we just got done with, and it kind of looks like we have a germinal center inside a follicle. So these are Peyer's patches. So this is what, I'll erase the red so you can kind of see it a little bit better. So this is what we're talking about with our lymph nodules, our collections of macrophages, T cells, and B cells embedded in the intestinal lining. We call them mucosa-associated lymphatic tissues, MALT. Well, now we know what the mucosa is. So here's the mucosa, and here's the lymph nodules associated with the mucosa in our digestive tract. And our tonsils were the same thing. They were embedded in the mucosa of our pharynx and our oral cavity. All right, and then the last one I wanted to show you, this is the colon. So this is a large intestine. And again, each one of these look wildly different from the other ones, but the point that I'm trying to get to you is we have a serosa, we have a muscularis externa, we have a submucosa, we have a muscularis mucosa, and we have a mucosa. So all 4 layers can be identified in each one of these slides, but they look very different because they're modified. So where in the stomach, we have a thick mucosa with gastric pits to secrete mucus. In the jejunum, we have a folded mucosa to increase surface area for absorption. In the ileum, it's folded, but not as much because we don't have as much absorption of nutrients that take place, but we do have some Peyer's patches. In the colon, we have kind of some folds, but if you're to zoom in, you may be recognizing all those little white guys like right in here, these little speckles. Can't draw very well on that. They're goblet cells. And what do you know about goblet cells? They secrete mucus. So in your large intestine, we are secreting a lot of mucus. We have to lubricate your colon as your food waste products the fecal matter is starting to move through, it has to be lubricated because we're reabsorbing water, it's getting solid. We need to protect this mucosa, but we still have all 4 layers in every example. And actually here we can see the 2 layers of the circular and the longitudinal of your muscularis externa. So I really like these ideas that even though we have this basic plan, we can see modifications to allow for the proper functioning to take place. So that's that structure and function again. Okay, so that's the histological organization, the layers, if you will, of your alimentary canal. All 4 are usually there. There's a few exceptions. And then modifications of those layers are going to help with whatever job that organ has to do to work on absorbing those nutrients, that big picture idea. All right, and then lastly, just again, some of this foundational information talking about the control of digestive functions. And again, this is just baseline stuff. We're going to get into specific examples as we go through, but there's 3 mechanisms of digestive function. We have neural, which neurons control. We have hormonal, we're going to be learning a whole new slew of hormones, and local. So back in the cardiovascular, we saw the same kind of thing. We had local autoregulation, and if that didn't work, we brought in neural and hormonal. It's the same kind of idea. So we have local mechanisms right here. I'm going to start with number 3. So our local factors, we have the lining of anywhere along your digestive tract can recognize changes in pH, physical stimulation like rubbing of food or substances on that mucosa, and chemical stimulation. So partially broken down carbohydrates or proteins or those types of things. So that can trigger local responses. So it can trigger endocrine cells to release locally, and it can also trigger secretory cells like buffer, mucus, other types of secretions in that local environment. Okay, so that is our local mechanisms. I'm going to jump up to neural. So we got to go flip a little bit back to the autonomic nervous system. So go back to chapter 16. I'll wait for you. Okay, welcome back. So chapter 16, visceral reflexes. You can go back and review if you wanted to, but remember we had long and short reflexes. Remember reflex, we had the receptor, we had the afferent neuron, the control center, the efferent neuron, and the effector. So those were our 5 components of a reflex arc. In autonomic or visceral reflexes, the short reflexes just went to the ganglion, was the control center or the processing center, then right back out the efferent neuron, where the long reflexes went back to the central nervous system and then back out the 2 motor neuron pathway. So that's what this is kind of showing. So in those plexuses that was sandwiched between your the submucosal plexus and the myenteric plexus we just saw 2 slides ago, you're going to have some receptors in there. There's going to be stretch receptors and chemoreceptors, and they can trigger neurons in the myenteric plexus. This is going to be your ganglion, right? Parasympathetic, controls your rest and digest. We're at the digest part, yes. So here, if we have something coming in, we can get a receptor here, go to an afferent neuron to the control center, efferent neuron out to your effector, which is peristalsis or segmentation, your muscular movements, or it could trigger glands to be secreted. So that would be a short reflex. On a long reflex, we would have the sensory receptor go to the myenteric plexus. Oh, sorry, that's not right. I gotta do my long reflex. So here's my sensory receptor. Instead of going to the myenteric plexus, it goes up to the CNS, which could be the spinal cord, or you could send an ascending track up to the brain. But in this case, from your CNS to your myenteric plexus, that's going to be your preganglionic neuron. Oh gosh, that would be bypassing the sympathetic chain, synapsing in a collateral ganglia, postsynaptic neuron going from the collateral ganglia out to the myenteric plexus, and then eventually communicating to secretory cells, endocrine cells, or muscular cells. So we can have a short localized response, or we can have a broader organ-wide response. So for example, local factors and short reflexes in the neural control work on a small piece of the organ. So for example, the small intestine, it's like 20 feet long. So let's just say we want activity just in the duodenum, which is like the first 6 inches. So we could get stretch receptors, local receptors, activating that part right there in the duodenum and leaving the rest of the small intestine alone. But if you're to bring in a long reflex, you might get a sensory, some kind of stimulus showing up in the duodenum, goes up to the CNS, comes back down, and then it will affect the entire organ system. So think short reflexes are for local control, long reflexes are like organ-wide control. Okay, so that's our neural control. And so now the last one is hormonal mechanisms. So there's quite a few digestive system hormones we'll be talking about. And these are just like our endocrine. They have a stimulus that's going to trigger them to be released. They have a target organ that they will go to, and then they will have an effect. And a lot of times they go via circulation for them to be a hormone, right? And then they come right back around right to their neighbor. Like you could have a hormone cell here, and it's going to get a stimulus, and its target organ is right next to it, but it's got to go through the bloodstream, come back around, and then target its neighbor. I know it seems kind of a waste of time, but that's what it is. So when we get to the hormones section, we'll talk a little bit more about those. So for this control of digestive function, know that we have some local factors that can cause responses. We can have neural short and long reflexes that can affect local parts of the organ or the entire organ. And we have endocrine effects that can be local or long body organ-wide, local responses or organ responses. And we'll see that when we get to the hormones a little bit later. All right, so that kind of wraps up this first introductory mini lesson on the big picture, the layering of the histology, and we're going to see kind of repeatedly those 4 layers as they get modified from place to place. And then lastly, how is digestion coordinated and controlled? And we're going to see these 3 topics again as we get into, say, like the control of the stomach or the small intestine or the pancreas, those types of things. All right, I will see you next time.